Alex share the screen? Like, is it working?
Yeah, let me try.
Thanks, Rachel.
Thanks.
See. Oh, wait, hold on. This isn't the right one. Sorry. I think it's my fault. Maybe I'm just a little bit challenged. 1 second.
No rush. Perfect. We can all see it.
Okay, cool. Is it okay if we start? Or did you say 7:05? I'm sorry if I missed
Oh,
that.
no, no, you can get started whenever.
Okay, okay.
I was like, give it a minute for people to join, but that's
Okay.
perfect. And then I'll send the slides out tonight whenever
Okay.
you guys are done. Yeah, thank you.
I can't see the chat, so hopefully you aren't saying anything to me there, but hey everyone. Oh, let me show my face. Hold on. Wait, I don't even know how to do that. Oh, there, found it. Here I am. Okay. My name's Rachel. Alex is here. Jenna is not feeling well. So I'm gonna start. And if you have any questions, just unmute because like I said, I can't see the chat. Yeah. So first one is amino acid metabolism. So for the first objective, it's kind of just memorizing, unfortunately. But ketogenic, the only one that's or only two that are ketogenic are these two. So maybe you can come up with some type of what do they call it? Mnemonic. If I came up with one on the spot, you could do like KILL, ketogenic, leucine, lysine. These are both. Um, And then this is just a definition of what glucogenic versus ketogenic is. So ketogenic is from ketone bodies, and then glucogenic, um, it can form TCA cycle intermediates. So it gets plugged into the cycle, and then as the cycle goes around, glucose is made. So just to understand what it means. Now, we all know Dr. Bog loves a chart. I don't love the chart. I think she tries to like put everything into one to make it easier for memorizing, but I sometimes used to get overwhelmed with that. So what I would recommend is go through the lecture slides where it's more detailed and have her like chart next to you. And then you can kind of see with words from the lecture slides and then compare it to the chart to see where it fits in. So I just included this because if I'm not mistaken, I remember her liking, you can see my mouse or no? Oh, it's okay. You don't have to unmute. But hopefully you can. But the substrate row or column, I would try to remember because I feel like she liked to ask the different substrates that could become glucose. So I would just try to keep up with that. This is unfortunately another memorizing situation. Um, my friends love to come up with mnemonics, so they would turn this like HILL, all that into like a sentence. And that helped them. For me, it was one more thing to memorize, so I didn't bother doing that. But maybe that would help you. Or there might be like a YouTube video with like a song or something to memorize it. Or Chat GPT. Um, so for me, like I said, she gives a chart. I kind of just put the words from what I gathered from the chart or whatever images that she had. So hold on, let me make this smaller. So asparagine is hydrolyzed by asparaginase, so that's the enzyme and then ammonia and aspartate are formed. Then aspartate can go on to form oxaloacetate by transamination. Glutamate and arginine can both become alpha ketoglutarate. I would know that for sure. She loves asking about alpha ketoglutarate. Production of arginine is in the urea cycle and the kidneys. And there's a different form that gets produced in the kidneys. That's why it doesn't use arginase like the other form in the urea cycle. Hopefully that makes sense. Another transamination reaction is alanine and cysteine, and then that forms pyruvate. Threonine forms pyruvate. Glycine to serine has a byproduct with tetrahydrofolate, which comes to be important later, and that also can form pyruvate. Phenylalanine can become tyrosine, but that requires tetrahydrop, whatever that word is. There's a lot of big words in this lecture. So one, excuse me. Second of all, try not to get them too confused because that was something I kind of struggled with. So tetrahydrofolate is different from tetrahydrobiopterin. There we go. All right. Oops, sorry. So this is the, what is it called? Objective, explain the cofactor roles of these things. So here are the big words again. BH4 is a cofactor for hydroxylation reactions of phenylalanine, tyrosine, and tryptophan. And then that also helps to produce nitric oxide from arginine. So that is like hopefully you know what a cofactor is, but it's like something that gets in to the cycle that helps push it along forward. And sometimes without those cofactors, it won't work, or most times. So I do think she likes to question those as well and the difference between these. So I would just pay attention. And then sapropterin is a synthetic version. So it's an actual drug that they can give if you're low on this. So maybe she might have a question. The patient is showing these signs and they gave them this drug, sapropterin, whatever, for what? Or why would they do that? Or what is it replacing or something like that? So she might ask that with that sapropterin. Tetrahydrofolate is the active form of folic acid, which folic acid, we're going to talk about that again later towards the end and how it helps to synthesize amino acids. It's important to know inhibition. I also believe Dr. Bog really likes asking about the inhibitors and the regulators and all those things. So know that methotrexate inhibits tetrahydrofolate. And then she might present a patient with spina bifida and then say, what is the patient deficient in? So it could be folate or tetrahydrofolate. So methionine is a precursor to SAM, which I would remember what SAM stands for because then that helps you remember what it is or like where it came from because it has methionine in the name. So breaking down of methionine produces succinyl-CoA. And these are the different things that SAM is important to. I don't know if she'll ask that, but sometimes I think I would get caught up in just memorizing the facts and not realizing that she could ask a question with a patient presentation. And that I know that that's not always how we are taught. So just keep in mind that SAM is important for myelin sheath, epinephrine. Yeah. So tyrosine can make melanin, dopamine, epinephrine, norepinephrine, thyroid hormone. Dopamine forms HVA, epinephrine, norepinephrine form VMA. definitely know that too much excretion of VMA, so you can say she might say from epinephrine and norepinephrine or whatever, can cause a pheochromocytoma. Definitely, she's probably going to ask something about that. Definitely, probably she'll ask something. Tryptophan, niacin, I think she really likes to know or to ask about. But then there's also Hartnup disease related to defective transport of tryptophan. So then the patient would present with tryptophan in the urine. They'll also have this red scaly rash. I just picked a different because the picture she has is the African American man with the neck that's like hyperpigmented. So I just wanted to use a different picture in case, she uses a different picture. Serotonin is found in the intestinal mucosa, and we know serotonin is for sleep and temperature regulation. It can be inactivated by MAO, which causes an increase in the 5-HIAA, which can lead to carcinoid tumor. So I know you're probably like, there's no words on here. There's a lot of words, but not a lot of words. But I think if once you go through the lecture notes, I would come back to this and then use it as a refresher. Don't let this be the only thing you look at. Or you could use it as a tool to tell you what is important to pay attention to. So antidepressants are selective serotonin reuptake inhibitors and MAO inhibitors. So inactivation of MAO, less serotonin, right? Yeah. Histidine can form histamine, and we know histamine is from mast cells when you have an allergic reaction, and it can affect your gastric secretions, and we know that allergic reaction means inflammatory reaction. So I think you just need to know that histidine can form histamine, and it's secreted by mast cells. That one I don't know if she asked that much about. So glutamate is an excitatory neurotransmitter, but it can also form GABA, which is an inhibitory neurotransmitter. So just know the relationship between those. I used to be like glutamate, my mate, my friend, we're excited to go into the brain together. So glutamate, GABA. I just came up with she's gabbing too much. You want to shut her. Okay, not shut her. Shut them up. Inhibitory. This is how you have to think to memorize these things. Arginine, I'm sorry, forms nitric
Rachel, you
oxide.
should tell them the song that you came up with to memorize everything.
And what's that song? I don't know the song, so
No, I'm just
you're telling
joking.
me something. Um, Arginine forms nitric oxide. Nitric oxide relaxes the smooth muscle. Glycine forms glutathione. Creatine, sorry, not creatinine. Bile acid, heme, and collagen. Oh, the heme and collagen were on a different slide, like with a table. So I put it in bold just in case so you don't miss it. And then these are the three things that make creatine, GAM or MAG. I don't know, however you want to remember it. It's reversibly phosphorylated. I feel like that could be important. Um, the kinase enzymes are phosphorylators, so you don't really need to memorize that. Creatine phosphate stores high energy phosphate in the muscles. That's why we have the dog who's muscular. And creatine is a marker of glomerular filtration rate. So if the kidneys are working, then creatine may not be up. If they're a little bit dysfunctional, then it could be up. And I'll come back for the next ones. This is Alex.
All right, perfect. Hey guys, I'm Alex. So I didn't really go over the content. Oh wait, Rachel, could you go to the next two slides? So I started going over content and then I was like, you know what, all you guys have to do is just memorize the slides. So I just came up with questions. So I mean, this is definitely, I'm looking at another monitor, by the way. So this is definitely going to be a question that they're going to talk about a patient with PKU and they're going to ask you one of these two, if they're like, which one's deficient, So it could either be phenylalanine hydroxylase or dihydropteridine reductase. So yeah, and then I just came up with like 4 or 5 questions on each objective. So here's the first one. I don't know if you guys want to put it in the chat or I could like wait a minute and then go over it. However you guys want to do it. So is everyone saying D? Chat. Yeah, D. Yeah. Yeah, so yeah, this one's D. Rachel, can you go next one, please? So I tried tricking you guys a little bit. I didn't put the classical one, which would be the top one, phenylalanine hydroxylase. So that one would get better with diet. So you could give BH4, which is like the coenzyme to make that reaction work. I'm pretty sure that one's in the metabolic map. So just another reminder to always be looking at it. But yeah, in this one, you can see that the bottom enzyme is deficient and that can cause PKU. So you can go next one, Rachel. Thanks. Okay. Yep. So everyone got C. Good job. Oh yeah, could you go next one, please? Thanks. So yeah, this one was C. Um, this is going to be another one that is probably going to get tested. So pretty much the end result is that melanin isn't being made, so that's what's going to cause the light skin. And is there anything else I have to say about that? Um, why not tyrosinase in this specific case? Why not tyrosinase in this? What do you mean? And Rachel, if you know the answer, feel free to step in too. Oh, sorry, I didn't see the question. Oh, okay. All right. Do you have a question? Do you? I can answer. Yeah, it is tyrosinase. All right. Looks like we got it sorted out. Um, here's another one, a little bit second order, not just like straight recall. So let's see how well you guys know the material. Got one for C. B. All right, so yeah, you can go to the next one, Rachel, please. So this one is clearly branched. So the key with this one is it's going to be branch chain amino acids. So those are leucine, isoleucine, valine. And I saw Shabab, you said it's nonpolar, which is technically correct, but it's not going to be the best answer because branched is one that better describes them all. So I think valine and leucine and I think isoleucine are all nonpolar amino acids. That is true. But the reason that this is wrong or that nonpolar is not the answer is because branched is like the more specific classification of all of them. And also you can see in the slide says branch chain amino acids. She's definitely gonna ask about, yeah, probably. Appreciate it. I got you, Shabab. So yeah, this one's a branch. So yeah, next one. All right, yeah, we can go to the next one, please. So this is actually a copy from my map. I went in the map, actually annotated it for this question. So yeah, this was like an easier one. Literally just look at the map. What do you think, Rachel? You could probably get like 10 questions right just from looking, or wait, I don't know how many questions they get, but I remember on our exams are like 75 questions. Probably 10 of them you could literally just get by looking at that. So look at the map, you'll get free answers. And yeah, so just to reiterate myself, I didn't make the slides to go over the content because, in my opinion, all you need to do is just memorize everything on those slides. So if you memorize everything on those slides, you will do well. We get 20, you get 24 for, yeah. So probably five of them, literally just look at the map. Anytime you're not sure about anything, look at the map. Look at the map and, maybe it could steer you in the right direction or it could literally just give you the answer. So if no one has any questions on my section, then I'm going to let Rachel take the lead again.
Okay, so the next
Yeah.
one is
And thank you, Rachel, for presenting these slides. Appreciate it.
no problem. Um, okay, so synthesis of purines and pyrimidines. So kind of wordy again, but it's just to have words to go with the charts that she makes. So de novo pathway, nucleotides are made from small building blocks from amino acids and other things. Whereas salvage pathway, you're literally salvaging bases from, it could be the breakdown of nucleotides or whatever. Sometimes the intermediates are formed from other, what is it called? Pathways. So salvage, you're getting nucleotides from that are just, let's just say, in the air, in the cytosol, whatever. De novo, you're making something new. You're building something new. Know that a purine is a pyrimidine ring with an imidazole ring on top of it, and it starts from ribose 5-phosphate. Liver is where you have de novo. I would definitely know the difference between that. So liver is where you mostly have de novo purine synthesis, and then it's transported through the blood. And then all the rest of the tissues, you'll use the salvage pathway. Hopefully that makes sense. Because the liver breaks down a lot of stuff. um, so yeah, so you're making things from scratch in the liver. And pyrimidine synthesis can be in any tissue. It kind of said it just wasn't specific. So I didn't bother putting, oh, actually, I think I have the picture on this slide, so we'll go back to it, but this is the actual purine pathway. But obviously, we know PRPP is the big dog. We definitely need that. That can be made from ribose 5-phosphate. So you're starting with ribose 5-phosphate, which comes from the HMP shunt, pentose phosphate pathway, whatever. So then you make, after PRPP, you make ribosylamine, whatever. And for a long time, I would get those mixed up. So that's why if you're going to use the abbreviation PRPP, then maybe you can use PR or PRS so that you don't get them mixed up. So the main or the rate limiting enzyme is the PRPP amidotransferase. So definitely know that. She loves the rate limiting steps. And no, I don't know if it's going to come up in this test, but just always know that there's a difference between rate limiting and committed. And so just make sure in case she tries to be tricky with the wording. IMP dehydrogenase is the target of the immunosuppressant drug mycophenolic acid, and that disrupts the B and T cell DNA replication. So then you have less GMP, which means you have less GTP. And yeah, so then, people use the mycophenolic acid for preventing graft rejection since it's like an immunosuppressant. So I just screenshotted this because there wasn't really a way to paraphrase it, but I would definitely know these things. PRPP activates, which just means pushes the cycle forward or the synthesis forward. Whereas ATP and GTP, GMP and AMP inhibit. They go back and inhibit, which we all know how that works. You're making a lot of the product, so the product goes and inhibits the path. And then know that GMP is used to make ATP, but AMP is used to make GTP. And yeah, so for all the pathways for this test and all the tests, you sometimes you don't have to know all the steps. I'm sure she said that to you, but you definitely need to know the regulators, the inhibitors, what's feeding back, what's not, and the like rate limiting enzymes. I'm sorry, I'm coming off of a cold. Okay, so salvage pathway for purines. Definitely no leash Nyhan. She loves bringing that up. So know that your HGPRT is not working as well. So that's how you get the syndrome. And self-destructive biting behaviors, she'll talk about, or whoever is going to make the question, is going to describe a kid who has self-destructive biting behaviors. And then she's trying to allude to leash Nyhan. Or she might say the patient has hyperuricemia. And know that PRPP is still in excess, and the products are not being made, which that makes sense, right? The thing before is the enzyme that's being inhibited is building up, whereas the product isn't. Okay. Kelly, I don't know Kelly, but hopefully she doesn't ask about it, but I put it in there anyway. But definitely know that if she is talking about HGPRT and she says self-destructive, no, it's leash Nyhan. If she doesn't mention that, then no, it's Kelly. I don't remember her asking about APRT, but I put it in there anyway. Just know kidney stones. That's kidney stones, right? I think so. Okay, de novo pathway, pyrimidine. So pyrimidine rings are from glutamine. I don't know if you can see my mouse, but I'm pointing down at the picture, and just remember that. I feel like you want to know the big picture from the pathways. I vaguely remember her asking the sources of atoms in the pyrimidine ring, so just know that. CAD is a like abbreviation for the first three enzymes of the path. Definitely know that dihydroorotate, whatever, is not in the cytosol. Pretty sure that was a question. UMP synthase, know that it has two different activities. CPS2 is in pyrimidine synthesis. You can think of pyrimidine has, oh, it has three eyes. Okay, never mind. Don't think of it that way. And this is also the UMP UTP line. That's literally just taking the chart that she had and putting it into words. And CPS2 activates or is activated by PRPP, whereas it's inhibited by UTP. So that's similar to the product inhibits the starting enzymes, whatever. Like Alex said, it is a lot of memorization, but so this is just a different way to read the material. So whatever, aciduria is due to two different things, and it causes two different symptoms. So in UMP deficiency, you have leukopenia and severe anemia. So this picture in the bottom is the hypochromic erythrocytes. So you see the red blood cells that have the big white circle, and then the megaloblastic bone marrow is the big purple cells. So that's only in orotic aciduria when it's a deficiency of UMP synthase. Whereas if you have a deficiency of OTC, you get an accumulation of carbamoyl phosphate, which that then spills into the cytosol, and that because there's a lot of carbon. Let's see if I can go back. Carboxyl phosphate here. You skip this step. Wait, can you guys see my mouse? Because then I'm just gonna be looking stupid. Just a quick chat, yes or no? Okay, perfect. So, oh Lord. Okay, here. I feel like the professors. Okay, so yeah, right here you get a buildup of this carboxyl phosphate. So you just start the path from here and you skip this step, which if you remember before, we said that was the rate limiting step. So you get a buildup of orotic acid and then hence orotic acid urea. And this is a hyperammonemia is only in OTC deficiency, not in the other one. And then you treat this with uridine. All right, so now we're making deoxyribonucleotides, and the big enzyme for that is ribonucleotide reductase. Thioredoxin is a cofactor. That's important. That might be on the map. If this pathway's on the map, then it should be on there. DATP is a negative regulator. So it inhibits. Hydroxyurea is used to treat cancer. I don't know how important that is, but I didn't want to not include it. And this is also a picture from the slide. I think she just wants you to remember those facts. Okay, antifolates. So antifolates can be bacteriostatic drugs, which means it stops the growth and reproduction of bacteria. It doesn't kill the bacteria, it's bactericidal. Yes, I agree with Alex that the map gives you a lot, that's for sure. And it shows the cofactors as like their abbreviations, like B9, but it shows in the bottom left what those things stand for. So just make sure you're using the map. I foresee her asking about the antibiotics, sulfonamides. I feel like a lot of the professors like that. And it inhibits this one right here, folate synthesis in prokaryotes. This one is antiparasitic. And then we move down to the anti-cancer drugs, and they interfere with nucleotide synthesis. By being incorporated, because so they're just like, they look like the nucleotides. So they're incorporated into the building of the DNA, or they inhibit the enzymes that are building the DNA. So then it affects it. Methotrexate is an example of that. I feel like she likes that one too. Know that formyl tetrahydrofolate is needed for purines, whereas methylene tetrahydrofolate is needed for this conversion. 5-fluorouracil. So this is an anti-tumor agent, and it binds permanently to thymidylate synthase, so that makes it a suicide inhibitor. And if we just quickly jump down to right here, sorry, it needs to be activated by the salvage pathway. But these two, FUTP and they inhibit two different things. So UTP inhibits RNA processing, DUMP inhibits thymidylate synthase. She's probably going to ask you about 5-fluorouracil. So just know that it permanently binds to thymidylate synthase. Cytosine arabinoside inhibits DNA polymerases for non-Hodgkin lymphoma. So she might present it with the non-Hodgkin lymphoma and then say like, well, what's going on? Why this is happening? The patient's being treated and what's going on, you know? So I know I used to like omit the clinical stuff because it's like, this is not that type of class, but she might put it in the question and that's your like hint in which direction she's going. So just keep an eye on that. So these are the last two things that interfere with the synthesis. Acyclovir, so that's for herpes. It is incorporated into the DNA chain and it terminates the chain from growing. So it stops DNA short, which obviously we know is going to have a impact. And then nucleotide reverse transcriptase inhibitors inhibit reverse transcriptase. So that's kind of self-explanatory. and that helps to treat HIV. So this is my last slide. That's why I put these GIFs or whatever, but I'm gonna read for Jenna. So Jenna's got catabolism of purines and pyrimidines. General tips. Use these slides as a guide for your studying. Some are straight from the lecture and have all the details. Others are a summary. So it's the same thing we both said. Make sure to go through your lecture notes, fill in the gaps, and focus on the red and blue text. I agree. I know you probably have heard over and over, just memorize the blue and red words. And sometimes that's good, but sometimes it's like, okay, stuff's missing. So you do have to read the stuff around it, but normally Dr. Bog is good for asking the things that are in red and blue. So digestion of nucleotides occurs in the small intestine. So sorry. 90% of purines are recycled by salvage pathway. 10% are degraded to uric acid or urate. ADA and PMP and xanthine oxidase are important degradative enzymes in the pathway. Xanthine oxidase. So, okay, uric acid is produced in the hepatic and intestinal tissues. Hydrogen peroxide is generated. In non-hepatic tissues, bases are not readily degraded and available for salvage. So that's what I said in the very beginning, that the liver is where you do the de novo, because uric acid and I guess other bases or whatever are being degraded. But in the non-liver tissues, bases are not being degraded. So you don't do, wait, I don't think I said that right. I'm not even gonna try to explain that. This is from the slide because I'm not trying to confuse anybody. See, you can tell I just researched my own slides. I did not look at these. So HGPRT deficiency, which we also discussed a little bit earlier, right? Increases hypoxanthine and guanine, leading to higher uric acid formation. So then you get hyperuricemia. I'm looking at the picture. If anybody has questions, I can try to answer, because I am just reading from the slide, and I apologize if you hate that, because I know a lot of people do. Purine salvage pathway. Salvage reduces the amount of PRPP, slows the de novo pathway. Yes, you need PRPP for the de novo. We talked about that already as well. And we talked about these. Remember, Leishman comes from HGPRT deficiency, and Kelly, I forgot her last name, but Kelly also comes from that. And then deficiency of this one gives you renal lithiasis. Okay, I'm reading. High level of uric acid in the body. This is what's considered high for each male or female. Causes of hyperuricemia, decreased uric acid excretion by the kidney, so kidney dysfunction. You could be in lactic acidosis, and then that could cause hyperuricemia, because if you have a lot of lactate in your blood, it's competing for excretion with uric acid. Could also get increased purine degradation, such as with HGPRT deficiency. You see, that's come up multiple times. So I would note that one, HGPRT deficiency. Myeloproliferative disorders, you get a high rate of cell turnover, so then you have a lot of purines. That could be from cancer chemotherapy. Von Gerke increased HMP shunt, leading to increased purine synthesis, because I can't remember what the R5P, right? Ribose 5-phosphate comes from HMP shunt, and then that pushes the purine cycle forward, or synthesis cycle forward. Fructose intolerance, increase in cellular AMP levels leading to increased urate. Lactic acidosis inhibits urate excretion because, like we said, lactate competes with uric acid. Excess alcohol intake. Oh, yes, because you get lactic acidosis when you have excess alcohol. And so once again, lactate is competing with uric acid for excretion, and you could get uric acid kidney stones. Gout. So you have a lot of uric acid. It's not soluble in water. Could it be competing for excretion with lactate for the third time? So then you get gout. So you get these sharp needle crystals in your joints, or you could have pseudo gout. So it's not the same type of crystals. They're not the uric acid crystals. They're calcium pyrophosphate crystals, or monosodium urate crystals. Is that the same thing? Uric acid crystals. Okay, I'm not sure. Causes also swollen tender joints. So different types of crystals can cause gout. Here's Leishman again, hyperuricemia. It's X-linked recessive. She could ask that. She could say the boy inherited it from his mother, and that could be a hint as to what it is. Don't forget the self-mutilation with the children. And then you treat with allopurinol because it's hyperuricemia, right? So that treats that excess uric acid, and then that could alleviate the hyperuricemia. Oh, xanthine oxidase inhibitor for buxostat, however you say that. Okay, so treatment for hyperuricemia could be a urosuric drug, colchicine, sorry. Decreases ingestion of uric crystals and decrease inflammatory cytokines, effective when given early. NSAIDs can decrease inflammation. We know that. Avoidance of consuming organ meat, so you have less purines, and alcohol, less competition with lactate and uric acid, and then weight loss enhances the renal excretion of urate. Okay, allopurinol and febuxostat, which we just said. ADA deficiency. So then you get the absence of both B and T cells. I agree, no SCID. She's probably going to ask what is the deficiency, why the child has SCID. You may not need to know the PEG ADA, but you probably should know bone marrow transplantation. Purine nucleotide phosphorylase deficiency. PNP uses phosphorylysis to split a nucleotide to free base and ribose 1-phosphate. Let's see, is that on here? Because that's something that maybe you don't have to remember. Yeah, it's on there. So if it's on the map, just remove that from your brain. Absence of T lymphocytes. Okay. So when pyrimidines are degraded, they can either create ketogenic or glucogenic intermediates. Uracil and cytosine are degraded to malonyl-CoA, which is ketogenic, whereas thymine is degraded to succinyl-CoA, which is glucogenic because it goes back into the TCA, I'm pretty sure, cycle. This is a table. I don't know if she found it or if she got it from the lecture. Oh, and that's it. Are there questions? This exam is going to suck. I'm so sorry. Thank you, Diego. Sorry guys if this was lackluster. I mean, we're on our last brain cell. We're gonna send them. Megna is going to send them to you. My typical advice for Dr. Bog is slides. I would go to her interactive slides and look at what she points out to be important. However,